Our oceans cover over 70% of the Earth's surface and contain more life than any other place on the planet. We know that oceans have a huge influence on the weather and the environment. However, they have also become a giant global conveyor belt of plastic waste. It's just over 100 years since the first completely synthetic plastic was developed. In that relatively short period of time, plastic has made its way into all aspects of our life. And while plastic has many advantages, its inability to break down naturally over time is causing big problems out at sea. Since the plastic age began, hundreds of millions of tons of plastic waste have been dumped in our oceans. And unfortunately, this figure is on target to increase tenfold in the next decade. As this incredible NASA video shows, the plastics have tended to collect in key spots around the globe called gyres, such as the Pacific Trash Vortex.
We can also see this in a computational model of ocean currents called ECHO-2. We release particles evenly around the world and let the modeled currents carry the particles. The particles from the model also migrate to the garbage patches.
The problem of plastic in our water is not confined to larger items. Researchers at the Galway Mayo Institute of Technology are investigating the relatively new phenomenon of microplastics.
Microplastics are small fragments of plastic. They can either be produced directly as small plastic particles, or they can arise from the fragmentation of larger pieces of plastic. It's in our clothing, it's in our food packaging, it's in our cars, in manufacturing of parts of cars.
Toothpaste, shampoo.
Yes, yeah.
Things that don't really necessarily need plastic in them.
Yes, sometimes those particles are doing no job at all, they're just colour. And, we could really do without that.
Microplastics, as the name suggests, are tiny particles of plastic and can be found in a wide range of unexpected places, from facial and body scrubs to your cuddly fleece jacket. In fact, when washed, the average fleece jacket can produce up to 2,000 plastic fibres. And even when plastic is recycled, it's often simply shredded into smaller pieces, which can also end up washed down the drain and ultimately out to sea. So what are the consequences of this for our seas and marine life?
The consequences are many. There's direct consequences for the animals that interact with those plastics. They can mistake them for food and eat them. Now that's kind of balanced by the fact that they excrete them as well. And also, if they're retained, these plastics absorb chemicals, or chemicals are actually put into the plastics during manufacture. Those chemicals can have toxic effects on the organisms as well. And that can transfer up through the food chains as well, with potential consequences for humans if it ends up in our food chain.
Research into the environmental impact of microplastics is relatively new. Dr. Amy Lusher undertook a major study to examine levels of microplastics in the seas around Ireland.
I moved to Galway 3 years ago to study microplastics and their distribution and implications on the marine environment, mainly because there hadn't been any studies previous to the research that we were going to do, and the North East Atlantic, there was just kind of this gap on a map.
So that meant you had to go out to sea?
It did, yes. I went to sea on the Celtic Explorer for a few months, and we found microplastics in almost every sample that we took at sea.
Are you serious?
Yeah.
Every sample?
Almost every, yeah.
There's microplastics everywhere in the North East Atlantic?
Yes. So in my research, we've looked at a group of fish called mesopelagic fish, and we found microplastic in 11% of the fish that we studied. We took out their whole digestive tract and dissolved it, so we knew that whatever we found in our sample was going to have come from inside the animal.
Are they fish that we ingest?
The mesopelagic fish, not at the moment. However, we did find plastic in commercially sourced fish from previous studies we've done. In the English Channel, we found 36% of the fish we looked at to have microplastics, again, in their stomachs.
It's scary. I eat a lot of fish.
It is scary because we still don't know the answers. It's a very new area of research. 10 years ago, we didn't know that microplastics were in the marine environment.
As researchers try to understand the impact of microplastics on marine life, the other question is, what can be done to turn things around?
I think we need to work a lot more on legislation and work a lot more on recycling and reusing plastics and trying to get away from single-use plastics and plastics that aren't necessarily required in our day-to-day lives.
The solutions are actually in our hands. We choose to throw away plastics the way we do, and if we change that behaviour, change the way we process plastics in the environment, we can actually really arrest this problem.
Our bodies are between 60% and 70% water. This changes at different times of our lives. A human fetus, for example, is around 95% water for the first few months, reducing to 77% water at birth. And in a 70-kilo person, there are 42 litres of water. But what happens when the water we drink is unsafe?
This is the second outbreak of cryptosporidiosis in Roscommon's water supplies in just 3 weeks.
It seems to be going on forever. We can't drink the water, we have to boil it or buy water.
Over 1,000 people are affected by the contamination.
Like, our water has been perfect and suddenly in the last couple of months our water has gone wrong.
It can be very inconvenient when you cannot drink the water that's coming out of your taps, but it can also be frightening when there's an organism in the water which can be fatal and which is defying most attempts to kill it. Cryptosporidium is a microscopic parasite that can infect animals and humans, causing them to become ill. It can be found in water, food and soil. In 2014, there were 394 cases recorded in Ireland. As part of her PhD at Cork Institute of Technology, Jennifer O'Leary is working on improving the diagnosis of cryptosporidium.
It can be fatal, especially in immunocompromised people, such as people with HIV or AIDS and people who have received organs from organ transplants, and it's also particularly nasty in children and infants.
Most diagnosis of cryptosporidium infection is done in hospitals using a basic staining technique. But Jennifer is working on a new diagnostic method which makes millions of copies of the DNA of the parasite.
And then this is detected by fluorescent detectors, and then you can then determine whether or not the sample is positive or negative. It offers a few advantages over the staining technique. First of all, it's more sensitive, so that means it's better able to detect low levels of the parasite in a sample. It's also more efficient because it's an automated process, and it's also more specific, so that means it's less likely to determine a sample to be positive when it's in fact negative.
Effectively diagnosing cryptosporidium is far more beneficial for patients and will save time and money. But is there any way of killing this parasite in our water? Cryptosporidium has a tough outer shell. That means it can survive outside the body for long periods of time and makes it very highly resistant to chlorine disinfection. There are currently 2 main ways to deal with it.
One is by physically removing the cryptosporidium using coagulation and filtration processes, so you're literally physically removing the oocysts from the water. And the other way is UV treatment, where the light penetrates the pathogen and inactivates it. So depending on the source and the type of risk that it prevents, then we'd install either one or both of those treatment types to make sure that we kill off or inactivate all of the cryptosporidium.
Two years ago, I visited an RCSI project in Uganda and saw how the simple power of the sun was harnessed to kill pathogens in the drinking water. It was extraordinary that leaving plain water out in the sun for 6 to 8 hours could make it safe to drink. Obviously, by comparison to this, our treatment systems are highly sophisticated. But here in Ireland, a researcher in Athlone Institute of Technology is working with UV light, which is up to 50,000 times stronger than the sun at point of treatment. Neil, you're trying to eradicate cryptosporidium.
Yes, we're looking at some new ways of delivering light, ultraviolet light, that has not been done before. Conventionally, UV light is used in drinking water plants, which is very effective. But this is a new way of actually enhancing the efficacy of it.
The difference with Neil's approach is to use pulsed light technology. Developed at AIT, it's an intense bright white light that's been enriched with UV to boost its antibacterial properties. It's then delivered in rapid pulses at up to 100 times per second, which kills bacteria and pathogens, including cryptosporidium. So the way that you're treating the water with the UV light pulses, could we see that with the naked eye?
No, you won't. If you look at the level of sunlight at the point of treatment, it's between 20 and 50,000 times intensity sunlight. So obviously, in terms of health and safety, it's not a thing you'd be doing.
Initial tests on the pulsed light approach have been positive, and full validation testing will be carried out before it's introduced into treatment centres. But Neil is confident that the days of cryptosporidium are numbered.
But we now can confirm, possibly for the first time compared to other gold standard methods, when you look at heavy fluences in the UV, like that we're actually, what we treat and what we kill is killed. As we're making sizable advances in the area of water management, water treatment, I think it's incumbent on us to also look at how we can translate those groundbreaking discoveries to our neighbours in Europe, and then also try and develop these technologies for other emerging economies. Water doesn't know boundaries. Microbes don't know boundaries. They don't require visas to go anywhere. From a humanitarian perspective, it's also important to share the understanding and the technologies with emerging countries, because it's the same water that we're connected to.
After the break, Fergus looks at extraordinary life at the bottom of our oceans, and Jonathan discovers why we need to care about the freshwater pearl mussel.
Light equals life, right? So scientists believed until the ocean floor gave up one of its deeply held secrets. In the 70s, near the Galapagos Islands, a team of scientists were exploring the seabed. All around, jets of hot black water gushed from the seafloor. Incredibly, in this total darkness, new life forms cover the seabed. But how can life exist without light? At plate boundaries, seawater enters the rock through cracks. As it travels through the rock, it heats up, but it also increases in pressure. This prevents the seawater from boiling in the same way that a pressure cooker works. At these high temperatures, up to 400 degrees, the seawater becomes acidic and leaches metals and salts from the rock. Eventually, when the seawater is forced from the rock at vents, it cools. This releases the metals and salts, giving the vents their characteristic dark colour and their name, black smokers. Special bacteria that live around the black smokers then convert the salt-rich fluids into life-supporting sugars. Up until then, scientists believed that photosynthesis, light leading to life, was the only way to sustain life. But here, the use of chemicals and salts, chemosynthesis, astounded scientists, opening their eyes to a whole new way of life. So, what do they call the creatures living in these extreme environments? Extremophiles. So sometimes in science, you just have to say it as you see it. See you next time.
When we look at a river like this one, on the surface, everything seems calm and peaceful. But underneath, there's a whole host of aquatic life struggling to stay alive.
One such creature is the freshwater pearl mussel. Ireland has one of the largest populations of this mussel in the EU, and they can live for over 120 years. As filter feeders, they're very sensitive to changes in water quality, making them useful as an indicator of the health of the ecosystem. But things have not been looking good for the mussel for some time. Changes in the way we do things like farming and forestry are affecting the mussel's ability to breed and the survival of their young. In South Kerry, the Kerry Life Project has been set up to restore the population of the freshwater pearl mussel in the area.
This river supports about 2.8 million mussels. The project also operates in the Blackwater River, that river hosts about 2.5 million mussels. The two rivers combined account for about half of all the Irish mussels, and that's a very, very significant portion of the European population, about 20% of all the mussels in Europe.
So what's been happening to upset the freshwater pearl mussel?
The big issue that we're seeing at the juvenile survival is very low, and therefore the population is in decline. So we're not getting a replacement of the older mussels as they die of natural age, with younger mussels coming into the population to replace them. So this population is likely to have gone extinct in probably the next 120, 30 years. So that's quite startling when you think about it. The big impacts on the mussel are the increase in levels of silt, which will come from agriculture and forestry, the addition of excess plant nutrients, which promote algae and macrophytes within the river, which compete with space, and changes in drainage cause change the behaviour and how the hydrology of the river functions. So you get more erosion during high flood events, and you also get, during low flow events, lower levels of water.
So, sediment runoff from land can have a serious impact on the freshwater pearl mussel. But how do you find out where the sediment is coming from? Researchers at Teagasc are using a novel technique to find out.
Well, what we're undertaking here is research in relation to sediment fingerprinting, or trying to determine where sediment in our watercourses is coming from.
You said fingerprinting. What do you mean by that?
Well, we collect soil samples, and the soil is a potential source of sediment. And each soil sample has a distinctive characteristic made up from a variety of different parameters, such as chemistry or the physical quality, the size of the soil structure, magnetics associated with it. The variety of these different parameters and variables will give that potential source its own distinct fingerprint. So in this case, this grassland field, along with other grassland fields, will have a distinctive fingerprint associated with grassland. Similarly, the soil associated with arable will have a distinctive fingerprint, and likewise forestry.
By taking soil samples from different types of land and comparing them to sediment samples collected in the nearby river, Dara can establish what the main sources of sediment in the water are. This lets him work out what land uses are generating the most sediment.
It's not the first place I would have thought you'd be looking for sediment in a river. You know, we're quite far from the riverbank.
Oh, a couple of hundred metres, yeah, but every bare soil is a potential source of sediments. So here we have a sediment sampler that's been in place for the last 8 weeks.
Kind of looks like a torpedo.
It does, yeah. And we're at the middle of, or at the end of a large agricultural catchment that's about 1,000 hectares, and all water drains through this outlet here. It comes through this sediment sampler. There's a small inlet, and then a larger size of the tube itself means that when the water comes in, there's a change in velocity, so the sediment falls out of suspension and theoretically should be caught now in this sediment sampler. So everything that has gone on in this catchment, the sediment will be now captured in this sediment sampler. There we go, so that's most of the sediment gone in. And we just wash this out a wee bit with river water and pour it in.
Having spent the morning getting my hands dirty, it's now time to analyse the samples here at the Teagasc laboratory in Johnstown Castle.
Helping me analyse my samples is PhD student Sophie Sheriff.
This is basically the samples that you took this morning from the river. So you can see it's quite a fine sediment sample. And then here's an example of one of the soil samples that you took. You can see that this sample is quite coarse. And this is kind of a problem with what we're doing. We need to make sure that we're comparing like with like. So one of the first steps after the samples have come in and been dried is to sieve these soil samples so that we can compare them better to the sediments that come out of the river.
Once we've sieved the sample, what next?
We will go for 3 different types of analysis, geochemistry, mineral magnetics, and also radionuclides. And this is how we measure the different traces. Now we'll get about 21 different traces out of the various measurements, and then we can piece apart the identities of each of those samples.
Sophie and Dara will analyse a number of samples at different points over the year to build up a full picture of what's happening in a catchment area. The samples I gathered today will help them understand what's ending up in the river.
We find that the sources do change quite a bit. So for example, in the summer, there's more vegetation in the channels. So that means that there's kind of protection on the riverbanks, which will stop the sediment coming straight in. And at that time of year, we might find more from the fields instead.
I guess the point of all this is that if you get to identify the source, then you can do something about it, right?
Absolutely. We can start developing and designing measures specific to that source area. So for example, if it's a riverbank, we can think about things such as riparian margins that farmers can be incentivized to implement in their landscapes.
The reduction of the sediment means that organisms like the freshwater pearl mussel can thrive.
Absolutely. Not only freshwater pearl mussel, a variety of different species, our insects, our salmonids, and also it's in the farmers or the landowner's own interest to stop sediment getting into the watercourse. Because often this sediment is nutrient rich, so they're better off maintaining the soil quality and maintaining the soil structure within their field or within their forestry, such that they're not losing it into the watercourse.
Figuring out how and why sediment builds up in the habitat of the freshwater pearl mussel is a major part of the Kerry Life project. If they can understand what's going into the rivers, then they can work with the local community to try to reduce sediment levels, which will ultimately improve the water quality.
Geochemical fingerprinting is a really exciting and novel approach to identifying where the sources of sediment are coming from in the wider catchment and how they get transported to the river.
The Kerry Life project will run for 5 years, and here's hoping that there'll be good news for the freshwater pearl mussel in the future.